Now, that's what happens over here. Now, there's something I want to just explain very briefly. The beta oxidation. All right? I just need more room over here. So, you're going to have carbon dioxide. Now, beta oxidation. We have something called beta oxidation. This happens in the mitochondria. And this is where your fatty acids, why the lipids actually could be made into ATP. Now, if you remember, if you recall, the structure of a triglyceride is basically a glycerol molecule and then three long chains of fatty acids, right? Remember that. Now, this can get broken up. The glycerol and then 3 fatty acids. The glycerol breaks up, and a glycerol, which I don't really want to get into, could actually be converted to glucose. Because there's carbons in it, okay? So that part can actually be put over here. But there is tons of carbons on here. There could be, let's say, 20 carbons on this chain, 20 on here, 20 on here. You got 60 freaking carbons on there. It's huge. What beta oxidation is that these fatty acids are able to get into the mitochondria. So now you have fatty acids. And the fatty acids get broken down by, if you look at these chains, I don't know if I told you, they're usually in pairs of, well, they're in even numbers. So what happens is 2 carbons at a time get broken off in here. All right? And what happens here is that it gets converted to acetyl-CoA. Through a series of reactions which I'm not going to go through. All right? What comes out of there is going, well, you got to put ATP has to be put into there, so you got to do that. But you're going to get, you're going to get, you're also going to get out of it NADH, so NAD plus, and you're also going to get, purple, you're also going to get FADH2 from FAD. All right? For every 2, so with the fatty acids, we're going to do it by 2s. How many colors here? So the fatty acids get broken down into CC, so 2 Cs at a time. And for every pair of carbons, you're going to get NADH, you're going to get FADH2. ATP has to be used, but you're going to get a whole, remember, you get 3 ATPs out of this, you get 2 out of this. Which these FADHs and NADHs be put into here. That's what this beta oxidation is. So we're able to take off the glycerol, make glucose to go through this system and may go into there. And the fatty acids can be put into this beta oxidation, which can also turn into acetyl-CoA. You know, things getting bigger? I'm more amazed at who the freak came up with this stuff. Because it's in this mitochondria that, you know, where it happens, I don't know how they did it. We just have to accept it. You think I'm boring, I gotta see these people. They're important, but yeah, how they came up with it, I'll never know. So what's going to happen here is that what your body's going to use up first to turn into energy is the carbohydrates that are floating into your bloodstream. Okay? Yeah, I'll talk about that. I was going to talk about gluconeogenesis. Right. So your glucose, which is the carbohydrates, they get used up first. Now, if you haven't been eating, you're fasting or on a diet, whichever, you're not eating and you don't have carbohydrates in your bloodstream, then the next thing that's going to happen is fatty acids. It's going to start breaking down the fats from your area, right? Your love handles. I'm pointing to mine. I don't want to point to yours, right? I got it. So you got your love handle. That's where the fatty acids are coming from, the triglycerides. So you got to use up all your carbohydrates before you can start losing weight. Does that make sense? And how it gets in there is what I just showed you. The fats will turn to triglycerides, the glycerol will be used into here, and these fatty acids will be used this way. I get more into the digestive system, as you can probably guess, but yeah. Okay? So let me talk about the beta oxidation thing. All right, so beta oxidation occurs in the mitochondria for your eukaryote. It occurs in the cytoplasm of your prokaryote. Like I said, prokaryote is too many, and they're making proteins. There's no compartment. Everything's getting mush-moshed in there, right? It's too much clutter. The process where it takes off 2 carbon pairs off of fatty acids, and they're made into acetyl-CoA. All right? This requires ATP to do this. Just what I said over here. That's why I think it's easier for me to draw it here and then just reiterate up here. All right? So you get NADH over here, and you also get an FADH2, which could be used to make ATP in the electron transport chain. Okay? How many ATP can you get from a fatty acid? Well, it depends on how long that fatty acid is. I mean, if it's only 4 carbons long, then you're going to make less. But if it's like 30 carbons long, you get a lot more ATP out of it. So it depends how much. All right? So, kind of like what I'm showing up there. The triglycerides, right? That thing I'm showing with this is triglyceride, right? The glycerol could be changed into, go through the glycolysis, pyruvic acid, and make acetyl-CoA that way. The fatty acids will get into the mitochondria and go through beta oxidation, and that will be converted to acetyl-CoA. Okay? It's busy. Yeah. Pretty much, yeah. It's when you need the energy. Because as soon as you make the energy, as I said over here, as soon as you make the ATP, you use it up. So what's going to happen is, and well, I didn't get into this part, I don't know if I'll get to it today, with fermentation, there's no oxygen. You're running a race. Right? We'll get into that. But yes, when if you're over here and you're breathing well and you've got enough oxygen coming in, and you haven't eaten for, let's say, a day or two, it's going to start doing beta oxidation. As long as you're getting oxygen coming into your body. If you don't have oxygen, like you're running a race and you're exhausted all your oxygen, then the pyruvic acid will turn into the lactic acid. But because there's no oxygen, it stays outside the mitochondria. Any other questions, concerns? Okay, so now let's talk about fermentation. The final piece of the puzzle. So we talked about with pyruvic acid, if there's oxygen, it's going to go to mitochondria. Now you understand this. Hopefully. Okay? And how you get energy that way. You get a lot of energy from there. Okay? Well, what happens if you don't have oxygen? What if you're running a marathon and you're using up all your oxygen? That pyruvic acid is not going to be able to make more ATP because you don't have as much oxygen as you had before. The amount that you're breathing in is not the amount that needs to be used. It's a lot less. So we have something else going on over here that takes place in the cytoplasm. And it's called fermentation. So the pyruvic acid, there's no oxygen, it can't get in here. Instead, it's going to go through something else. This is called fermentation. We're going to actually change it to a chemical called lactic acid. Or it's also called lactate. There's an H difference, but for our purposes, any place you see lactate or lactic acid, it's the same thing for this class. Okay? You don't make any energy on here. That's a key point. Instead, it's going to take this that's just made over here that isn't able to get in here for whichever reason. We can actually recycle this. So what happens here is that this NADH, through fermentation, will be oxidized to become NAD+. So where does all this NAD+ come in to make this? Because of fermentation. It's putting the NAD back into the system. Okay? There's a lot of steps in there. Don't worry about those steps. Now, we make lactic acid, but in other organisms, like yeast, they have the glycolysis over here, and when they don't have oxygen, they go through fermentation, but it doesn't turn into lactic acid, it turns into ethanol. The same stuff that's in beer and wine. And if you, like my father makes wine, he needs to put yeast in there to give it the alcohol in it. The yeast, without oxygen, that's why it needs to seal it, so oxygen's not getting in, it turns into ethanol, alcohol. But we can't. Can you imagine if we're running a marathon and people are getting drunk just for running a marathon? Not a good thing. So it turns into, instead, it turns into lactic acid. Acid, it burns. Ah! That's what makes you tired, is lactic acid. Okay? Now, the lactic acid goes into the bloodstream. So lactic acid goes in the blood. And it's then going to go into our liver. Now it's in the liver. Okay? Now what's interesting about this, the lactic acid could actually be changed back. So we can actually take the lactic acid in the liver change back to pyruvic acid. And then that can actually turn back to glucose. Now, if you don't need the glucose right now, the glucose is then going to be stored as glycogen, right? Stored starches in animals. And a lot of it will be stored in the liver. Long chains of glucose. Or if you need it, then what's going to happen is this glucose is going to be put back into this system. How it all interacts, it's like a spider web, right? There's one other part of this piece that I can explain is how you go from pyruvic acid to glucose. Well, draw the right this. There's just a word, and it's written up here. It's long. Just note that the star here is here. And it's called, just hear me out first, gluconeogenesis. Alright, so it's here. Now it's not here, it's there. And what it is that you can actually make glucose from non-carbohydrate sources. Like pyruvic acid is not a carbohydrate. But we could change it into glucose by way of this thing called gluconeogenesis. Gluco meaning glucose, genesis meaning synthesis, neo meaning new. So you're making a new synthesis of glucose, not from glucose sources. We could actually take amino acids, certain amino acids, we could actually change the glucose. See, amino acids aren't carbohydrates, but you could change them. And it's beyond the scope of this course. But that's what happens here, is that you're taking non-carbohydrate things and turning them back to glucose. I hope you're seeing that glucose is the major source that you need to get energy with oxygen. Glucose is the thing that's needed, sucrose will get changed into glucose. A lot of things get changed into glucose, but it has to turn into glucose. Okay? Does that make sense to everybody? That's about as far as I want to know what gluconeogenesis is. And this whole process, this process of fermentation going into the liver, this whole process over here is something called the Cori cycle, which I'm not going to ask you about. It gets really, really busy. Yeah, so what's going to happen is, and again, I'm not going through the steps on here, there's a lot of steps, but it's taken the NADH that's here, and we've got to get more NADH pluses for this to happen. So how does it do that? If there's any NADHs that are left here, this fermentation is going to take that, and one of its steps is going to get rid of that hydrogen, and is able to oxidize it to become NAD plus. So you have this cycle. This will convert it to NADH. This will convert it back to NAD plus. So it's this big cycle that happens over here. And it's all in the cytoplasm. All of this, now notice that there's no ATP matrix. So here's a little tip for you that I think students get mixed up. Anaerobic respiration. It makes 2 ATP. It's not coming from fermentation. It's coming from glycolysis. So if there's a question on the test that can ask, how much does fermentation and anaerobic reaction make ATP? It doesn't. Glycolysis is where you're making the 2 ATP, net-wise. There's no ATP that's being made here. The only purpose of fermentation is to recycle this NAD+, and the lactic acid can get into the liver, where later on we can make glucose through gluconeogenesis. Gluco meaning glucose, genesis means birth, and neo meaning new. So it's new birth of glucose. It's coming from some other non-carbohydrate source, a non-sugar source. It'd be neat to go from sucrose to fructose to glucose, because they're all rings. They're all basically descend from the same family. But you're taking an amino acid, you're making amino acids that have nothing to do with sugars, and you're making into a sugar, you're making a new formation from a source that has nothing to do with sugar. So that's what it is, a new genesis, a new birth of glucose. So let's just do the slide that go with this. Fermentation. All right? So after glycolysis, so everything I just explained is going to be now up on the PowerPoint. So after glycolysis, if oxygen is present, then the pyruvic acid will enter the mitochondria and go through the citric acid cycle and make your ATPs. If there's no oxygen present, then the pyruvic acid stays in the cytoplasm and goes through fermentation. So fermentation occurs in the cytoplasm, uses no oxygen, and keep in mind it makes no ATP whatsoever. I think that's confusing. I think students think that it does. It doesn't. So it's able to recycle the NAD+, as I showed you, and it puts back in the system so that we can put it into glycolysis. So this is showing you, here's glucose going to pyruvate, glycolysis. If there's oxygen, it's going to actually get into the mitochondria, and it goes through the citric acid cycle and the electron transport chain and all that. Or if there's no oxygen, it goes through fermentation. Pyruvic acid is converted to lactic acid or lactate in animals, but it can turn into ethanol or ethyl alcohol in yeast. So there's alcohol fermentation and there's lactate fermentation. And this whole process is going to make NADH get oxidized and become NAD+, and allows it to recycle for glycolysis. Right? So lactic acid fermentation, what I showed you here, is when our muscles get tired with lack of oxygen. The pyruvate accepts electrons from the NADH, and the lactate and the NAD+ are then made. So when fermentation happens, NAD+ is being made, and lactate is being made, or lactic acid, right? So as our muscles get more oxygen, lactate changes back to pyruvate. When you rest, it changes back. The lactic acid enters the blood and then the liver. Okay? So You have the glucose, goes through glycolysis, and makes 2 pyruvates, right? 1, 2. All right? If there's no oxygen, fermentation takes up, and that converts NADH to become NAD+, and you'll get lactate. You get 2 because there's 2 pyruvates here. Okay? Alcohol fermentation, this is what happens in yeast. All right? Pyruvate is converted to acetaldehyde and also carbon dioxide. So you don't get carbon dioxide with here. But in alcohol fermentation, yes, you do. All right? That's where, like the champagne, if you've ever had champagne, not that I'm encouraging that, the bubbles, it's carbon dioxide. Okay? So this accepts electrons from the NADH, and ethanol, alcohol, and NAD plus are made with carbon dioxide that gets released. All right? There's other fermentation pathways, which I'm not going to hold you to remember, but just to give you some idea, other things, instead of using alcohol and lactate, there's other creatures that make things, and we can use them to make soy sauce, tofu, yogurt, cheese, a bunch of other stuff. So if you have an interest in this kind of stuff, in food processing, and that's a big market, you'll have a job security, you're going to have to know a little bit of chemistry. Okay? Prokaryotes that rely on fermentation, we also have that in our intestines. Produces different gases, methanol, and that's why you let out the gas that way. All right? Because of those wonderful bacteria. All right? So alcohol fermentation, again, 1 glucose and 2 pyruvates, they go through fermentation, but unlike the lactic acid, you get 2 ethanol and 2 carbon dioxide that come out of there. But the NAD is getting recycled. All right? So alcohol fermentation, or just fermentation in general, is less efficient than cellular respiration. It only makes 2 ATP. The aerobic thing, and it's not coming from fermentation. These 2 ATP come from glycolysis, not fermentation. Right? Keep that in mind. It's coming from glycolysis. There's nothing here that's coming out. There's no ATP. Okay? So cellular respiration, on the other hand, makes 36 ATP. So look what's happening here. Here's the net wise. 1 molecule of glucose. In anaerobic respiration, you're only going to get 2 ATPs made, right? 2 going in, but 4 going out, net 2. But if we have oxygen, then this goes on, and with 1 molecule of glucose, you're going to get a whopping 36 ATP over here. Does that make sense? So, let me ask you this. This is how you're keeping track. If I said to you, in anaerobic respiration, how many ATP do you get net? 2. Now, be careful with this one. If you do have oxygen, then what's the most ATPs that you can get with everything? 30. Who says 30? Who says 32? 34. 36. 38. You get 38. Why? Because if you have oxygen, I want to know the total amount of ATPs you can make. You get 36 over here, but you get 2 over here. You see the difference? You've got to keep up and keep track of these ATPs and where they're being made. And you got to read the question carefully. If I said an anaerobic process, how much you make? 2. All right? Is that clear? That's why having oxygen is so much better for everybody. You get a whopping total 38 ATP. Give or take, it depends on how much hydrogen and stuff, but what we're going to use is 38. It varies between 34 and 38, but we'll say 38. Okay? That's what most books are saying. All right? Facultative anaerobes in some organisms can switch between fermentation and aerobic respiration. We're not one of them, all right? It uses fermentation if the electron acceptor O2 is not present, all right? When you get further into biology, you'll learn about those other anaerobes.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvic acid is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. 2 common types of fermentation are described here. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate, lactic acid. Let's see the relationship between glycolysis and alcohol fermentation. Glycolysis produces NADH, ATP, and pyruvate, pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In alcohol fermentation, the pyruvate, pyruvic acid from glycolysis, loses 1 carbon in the form of carbon dioxide, and the product is then reduced to ethanol by NADH. With the formation of ethanol, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Alcohol fermentation occurs in yeast cells. Let's observe the relationship between glycolysis and lactic acid fermentation. Glycolysis produces NADH, ATP, and pyruvate, pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In lactic acid fermentation, the pyruvate, pyruvic acid from glycolysis, is reduced to lactate, lactic acid, by NADH. With the formation of lactate, lactic acid, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Lactic acid fermentation occurs in muscle cells.
Okay, so let me talk about gluconeogenesis very briefly about this. I'm not going to hold you a lot, but you should have an understanding of what it is. This occurs in the liver and actually 40% of the kidneys. Okay? And what it does is it makes new glucose from non-carbohydrate sources, from glycerol, Remember how I showed you how triglycerides are broken up and the glycerol becomes glucose, right? That's gluconeogenesis. Taking amino acids, pyruvate, lactic acid. These are not sugars, so therefore these can actually turn into sugar. We have processes that do that. If you're interested, you could go get a different book and then go to a different class. I'm not interested for you to know those steps. Okay? And this needs oxygen to do this. All right? Occurs when blood glucose levels are low and the body's carbohydrates are also low. And it has to go through this. It's basically the opposite of glycolysis. You're going to go from lactic acid to pyruvic acid to glucose. All right? So, basically, glucose, well here, non-carbohydrate sources make glucose by way of gluconeogenesis. The storage area in the liver of long chains of glucose is glycogen. You learned about that. They're polysaccharides. To go from glucose in the liver and make glycogen, that's something called glycogenesis. If we're going to take the glycogen and break it up and put glucose in the blood, we're going to break it up. That means it has to have the suffix lysis there. So it's glycogen, how do you say that? Glycogenolysis? Okay. All right? But if you look at the words are going to help you. If you know what lysis means and gluco and stuff, the words are going to help you.